Okay, well, our second speaker is Zachary Underwood. He's an associate professor at BYU, and I think he's been, there 7 years now, or? Okay.
All right, so I'm going to follow up on a little bit of what Bruce talked about. First, I want to just acknowledge some of my other co-authors. Tate Campbell and Debbie Rigby and Jordan Bybee were graduate students on this project with us. So as we talked about, this idea of shredding whole trees is something that we do, or a management practice, to stem catastrophic wildfires on these ranges. The one thing that's really amazing or unprecedented about this is it's taking whole trees and shredding them in place, depositing them on the ground, kind of almost exactly where they existed before. And adding this amount of debris changes a lot of ecosystem function that we don't necessarily have a great handle on. And really, we're using the same sites that Bruce talked about. These are 31 different PJ sites located in Utah. They have a range of tree density from about 15 to anywhere to 40% cover. They're scattered all throughout the state, which is a wonderful thing. And we wanted to really, we kind of modeled our sampling of different grasses and soils off of sage steppe. So we have all the sage steppe sites that are included, but we've also expanded that to quite a different number of sites around the state. And really, we talked about some general changes. Bruce talked about perennial grass cover going up. We wanted to really look and identify if there was individual species relationships with this formation of the shredded material. What's going on with individual bunch grasses besides just Bromus tectorum? And also looking at what is it doing to the microbes. You know, the microbes, the bacteria, they do so much for us, but sometimes they're those unsung heroes in all of these ecosystems, where we just sometimes take them for granted. So we just wanted to understand what was happening to them, and also what was happening to specific plant species. And the way that we did this, we looked at three perennial grasses in general that were ones that cohabitate with Bromus tectorum, have some hypotheses out there that are generated that have competitive exclusion principles with Bromus tectorum. And really, we looked at Elymus elymoides, Poa segunda, and Pseudoroginaria. So, and also across these, the way that we did measured grasses was, again, standard sage steppe. It was line intercept. But what was nice about the shredded plots and being shredded, we could actually look out there and say, you know, here's where a tree was, here's where that edge of that tree was, and here are these interspaces that are usually filled up with other perennial grasses or shrubs. So we really, when we went out to these sites and when we first thought about, you know, how is shredding going to affect soil processes and in turn affect plants, we really thought that tree islands would play a big role, right? That these tree islands there are something you can see across the landscape. They could definitely change a fair amount of water regime and nutrient regimes. And so we thought they would really play. And we also thought that nutrients would be one of those big factors that microbes are controlling that are going to influence perennial grasses as well. And lastly, we thought that stuff that was happening that shallow surface, right? So when things get shredded, they lay down oftentimes on duff, a thick layer of needles, and are not necessarily in contact with that mineral soil. But we thought anywhere where we are contacting on the edge soils, where we're getting that interactions, this new woody debris onto these soils, they would have a big difference. So we looked at shallow soils and also deep soils, so deep soil processes as well. And we used a series of mixed models. This is just a nice way to utilize different covariates. Since this was a post hoc study, these treatments had all taken place. And we found with the NAPE imagery that April helped put together, we found out areas that had been treated and coinciding areas that were untreated. So we had this kind of paired study in all these sites with tons of sub-replicates. And we also had treatments that occurred over the 8 years, right? So you had to kind of take into account that, hey, these treatments occurred at different times. So we had time as also a covariate, and also those microsites as a covariate as well. And when we thought about these mixed models, we thought about there's three likely outcomes that would occur after fine woody debris was added or shredded on top of these soil surfaces. It was one, that soils all in general, this first one up here on A, that soils mediated everything that was happening from this fine woody debris and in turn was influencing grasses. The second one said that soils partially mediated some of this, but then there was also other things that we didn't really necessarily measure in this associated with soil processes that was also affecting this grass frequency on these plots. And last, that fine woody debris affected soils, but soils in turn really had no change on grass frequency. So these were our three outcomes. And these are our major modeling results. If you look at, we have Bromus tectorum and then our three perennials. In the case of all our species that we studied, except for Poa segunda, treatment of shredding increased those grass frequencies by about 12 to 16% on the landscape. And you can see this. So these are all what's called modeling intercept values. The fact that they're all positive, except for Poa segunda, shows you that there was an increase in this intercept. And again, this was about 12 to 16%. Poa segunda had no really sensitivity to shredding. It really didn't change in its distribution. And then what we did was, in our first set of models, we just had site as a model. And then we added site plus our soil variables associated with it. And only for two of our grass species, Bromus tectorum and blue bunch wheatgrass, did we see any kind of soils mediating the effect of these grasses. Elamus elamoides, although even after shred, you can actually see that the frequency of it actually went higher, and there was no real reduction in the explanation or the variance that we were looking at with including soil variables. So it's those two basically with pseudoerosionaria and Bromus tectorum that variability was reduced when we added these soil variables into the model. This is kind of 4 mixed models all added together to kind of show an overall representation of what we saw. And the first time for this side, I want to concentrate first on nutrients. So we had phosphorus availability and N mineralization rate. And as most of you know, N mineralization is that amount of ammonium that's being produced in these soils over a certain point of time. And even though we saw a huge increase in phosphorus availability with shredding, and this was mostly just in the surface soils, that affected actually none of the grasses. You know, we thought that phosphorus is pretty limited in these environments. It's going to help. It really did, to our surprise, really nothing. And actually, it negatively affected Poa segunda. And the way you can look at this is if you have your fine woody treatment, and again, this is by difference, by the control in the shredded plots, you would see if there's a positive, there was an increase in that variable, where you're going, so fine woody debris to phosphorus, there's an increase in phosphorus, but then you see that there's a negative value going from phosphorus to that grass, which actually caused that reduction. For Poa segunda, there could be something associated with a competitive advantage of this grass being able to exude some root exudates that help mobilize phosphorus, and when phosphorus gets higher and higher in these soils, or it gets more available, that competitive advantage of Poa could go away. For our two grasses that were modified by soil processes, this N mineralization rate in deep soils, and we're not deep soils. These soils are shallow. We're talking about 12 to 17 centimeters depth. Our shallow was 0 to 5. This was 12 to 17. This had the biggest impact on our grasses of any of our soil processes. So the more N mineralization that was occurring in deeper soils had a bigger impact on pseudoerosionaria and Bromus tectorum. We also kind of had this when we first hypothesized, we thought nutrients would be king, and then there's these other processes that we measure, like soil respiration, how active the bacteria are, how they're breathing CO2, just like how we eat food and breathe CO2. And we also looked at just the biomass. You know, is there any kind of just these coarse measurements of microbial communities of biomass? And what we found was that sure enough, when we were looking at deep processes, so like deep biomass, there was a deep microbial biomass, there was this negative effect associated with having more biomass being stimulated by the shredding itself, and actually having a negative effect on mostly on our perennial grass. So Bromus tectorum was just sensitive to N mineralization, but when it came to changes in our biomass and our lower respiration rates, it was pseudoerosionaria that was the most impacted. The other one was kind of a decrease in deep soil respiration, and this idea that the bacteria after shredding are not doing as well. You put all this coarse woody material on the soils themselves. They're eating up this recalcitrant carbon, and they actually have a depression in how much CO2 they're producing, and that could in turn affect other cascading effects that bacteria really work on. One of the big ones that we love in these environments is bacteria produce a bunch of snot or exopolymeric substances that hold soils together and kind of help retain water. And we really didn't see that going on in these systems. The other one to point is microsite did not matter in any of our sampling. It didn't matter if you were on the edge of a shredded pile, or if you were in the shredded pile, or if you were on the outside in the inner spaces. When we incorporated all of our data for all of these 31 sites, it was not significant. Time, though, was always significant. So, and we kept that in our model. The last arrows that you see on the bottom, there were other explanatory variables that we did not capture that was moderating or mediating what these bacteria or what these plants were doing. And I think the big one that is what Bruce said before, that if you put together end mineralization, if you have good end mineralization in deeper soils and you have wet soils, you're gonna have a higher amount of not only perennial grass but also of the cheatgrass as well. So it was beneficial for some of our perennials. They grew more, but it was also beneficial for cheatgrass. The one thing we did notice in time, and I don't have a great slide up here, but when we looked at the time intercepts itself, for all the perennial grasses, it was a positive relationship through time. That meant that for Elymus elymoides and blue bunch, as time progressed, their frequency actually increased through time, where bromus was negative. And so we actually had a decline over those 8 years of in this relationship of this model. All right, so let's talk about bacteria. And we still went through the same thing. We had our microsites, and we thought, microsites are going to reign supreme, again, because it has all this amount of woody recalcitrant material that's being deposited on these soil surfaces. And how is that impacting soil microbes? And I know, I don't know if many of you know, but if you take a little bit of soil, like a gram of soil in your hand, there's thousands and thousands of species of microbes in that little gram of soil. And the diversity of it is just immense. But we wanted to look at this as an unprecedented study where we're putting this coarse woody debris on the landscape, and let's see what was going on. And the first one that we found really was that just like we talked about, your shredding material you're putting on the landscape, this coarse woody debris, if you look at the bottom here, as far as it's putting in a bunch of dissolved organic carbon. So oftentimes when we go out and measure shredding studies that we did in the stage step, it was total carbon, total nitrogen. But the dissolved organic carbon is what bacteria see, right? They're solution feeders. They want to know what's in solution and coming to them. Well, after you shred, there's a bunch of more DOC, this dissolved organic carbon that's available for them. However, when you look in that next graph, there's this is how much microbe biomass there is per amount of that DOC. Even though that there's more DOC, that DOC is a poorer quality and is supporting less bacteria. And then at top, this is called microbial efficiency, and it just means that it's showing that it's even less efficient for these bacteria. So the way it is this is measured as a CO2 amount that's respiring per unit of microbial biomass per second, and it means that for them to convert into biomass, they have to breathe a lot higher and produce a lot more CO2, so they're wildly more inefficient than than a bacteria that's living in the inner space. So when we shred, it's bad for bacteria, even though we think, it's increasing carbon substrates and availability. These are network co-occurrence models that show how these bacteria are interacting in these soils. And they look crazy, but if you look at canopy control, canopy shred, this is edge control and edge shred, and that inner space shred, each one of these dots in these models are individual bacteria species. And they're not all the species that are there, but they are the ones that are significant. And they're the ones that have had significant relationships with each other. So all those dots are individual species, and then all those lines are significant interactions that these bacteria are having with each other. So it's bacteria communicating with each other, influencing each other in a negative or a positive way, but working together as a community. So it's kind of symbolizes all the different community interactions that we see. And the different colors relate to these different phylum or groups of bacteria. And what we found was that when we ran these type of simulations, there was a breakdown of this community structure from when you shredded material and put in more of this material on the surface. So if you look at the top, so you have a canopy control. There's tons of these interactions. When that canopy was shredded and some of those interactions started to go away over time, right? The shredded material was breaking up some of these connections, although in the canopy sites, compared to like edges, there were many more connections. And I'll show you this on this next slide. We run a bunch of statistical analyses on kind of explaining these communities, but I just really want to highlight two. There's one, if you look at this edges, this idea that these are the significant number of connections, that in these control plots, there was 14,000 interactions between these bacteria. And when this was shredded, it was cut in more than half. It was almost a third. So that there's a breakdown. Even though they're complex and they're still interacting with each other, edges are a point of confusion for bacteria, right? They don't know what to do with it. We usually have this idea of intermediate disturbance, and I actually thought when we first started doing this study that bacteria would be the happiest at the edges. You know, they got the best of both worlds. They got some inner space. They have some canopy. It was definitely not that way, even if it was shredded control or a shredded inner space. And last, if you see our inner space shred, it looked the same for inner space control, but basically you have all of these connections that are again happening in the inner space. So it's a more predictable area in these inner space soils for these bacteria to interact with each other. The other one that I want to show you is this idea of modularity. And modularity means bacteria that are focused or grouped together in these tightly knit groups. And what is kind of strange is that in these edges, though, these bacteria are more modular, which means they're more, even though you can't really see it in there, or you can, but it's this idea that they're more modular, that they're all depending on each other. And oftentimes we think of modularity in bacteria communities as bacteria that have gone through some type of disturbance and are now resilient or resistant to change. So I think these bacteria, again, in the edges are being, again, exposed to, although you think it's the best of both worlds, it's an unpredictable environment. Last, these are the genes that are associated there or that are expressed by these different communities. And these are called triplots. You have edges at the top, you have canopy, and you have inner space on each side. You have control, and you have that shred treatment. And what happens is we use a bunch of predictive tools to say, here are the bacteria that are there. What type of genes are they expressing? And in all cases for our canopy sites, the ones that are right under the canopy themselves that have those tight networks, they are producing about 236 significant genes that are significantly different from inner space or edges sites. And these genes are mostly associated with the breakdown of woody material. What's crazy, though, is those genes that are being broken down in the shredded and also the control, although they are focused on that recalcitrant material, they are very different from each other. One is more kind of polyphenolic, associated with needles and duff. The other one is a little more associated with breakdown, heavy lignocellulose material that you would see in that shredded component. Inner spaces, they did have a significant number of genes. They had about 66 genes that were significant, but that's a far cry from 236. And for edges, there were really no significant different genes that were being expressed by these confused communities that have very little interaction. So really, I think in conclusion, this idea that although we thought that shrub islands would be important, time is a big integrator or really important for shredded studies. Also this idea that deep end mineralization is one of those things that are really important. And we thought about, where does this deep end mineralization simulation come from? You have shredded trees, the tree material sits on top of that soils, but you have a lot of roots that are also decomposing. And sometimes these are a little more labile and a little less recalcitrant. And our ideas were that, although we didn't study roots, we think that it could be two things. It's either translocation of some more substrates down through the profile that are increasing the end mineralization deep in deep soils, or it's this woody material that is being broken down. And I don't think we have a good understanding of what's going on below ground. For a lot of our shredded studies, it's easy to focus on those surfaces. From what we've seen is there's a lot of stuff that's going on deeper in that soil profile that's important, along with water and understanding better water regimes. And then for bacteria, they're different from plants. They're the ones that are controlling a lot of these processes that we see, but shredding is confusing for them, right? They're less efficient. They're breaking down to their community structure. They're switching to something else, and it's not necessarily that it's bad. It's just something that it actually does exert some type of change, and you see that in the processes and the assemblage that they have, and also their gene function. And that's it. Thanks. Oh, let me just thanks Sage Step one more time, and Joint Fire Science for funding this research. So. Go ahead. So the question was, did we look at the fungal component of the soil microbial communities? Really, we did not. We did mostly bacteria. When we did this study originally, this is all next generation sequencing that we did on 16S ribosomal RNA. And when we did this, even just 2 years ago, the primer sets available for fungi were very few and really underdeveloped. That's changed now. If the study was now done in my lab now, we would have looked at it. Some of the points, the deeper depression that we saw with microbial biomass, that microbial biomass absolutely could have is fungi and also bacteria. So it would have been great for us to separate, what specifically in that microbial biomass was driving it. Was it changes in mycorrhizae structure that was, influencing that grass performance? But no, we didn't know for sure. So that was a certain, you're talking about a certain size of the coarse woody debris to maximize inputs. In the world of microbiology, anytime you can increase that surface area and decrease the size, it's going to be better as far as things we're breaking down. And in all of these, even in that model, the stuff for the bacteria that I showed you, that had Piñon and Juniper as well, like the different species, but we really saw no effect of difference of these drivers on bacteria depending on Juniper and also Piñon woody material, which actually decomposes quite differently, and we thought would have a much significant effect. But again, if you make it small, the bacteria have a more surface area to get into and break that down. So it's a good question. Go ahead, Bruce. Yeah, absolutely the same way. So if in the edge and the control, and actually edge shred got a little better. There was just, I don't think they could predictably say what they were getting for carbon inputs in those areas versus when you go out in the interspace and you have some shrubs and grasses. And our interspaces were all about, at least 3 meters away from the tree itself. So. Can you repeat that one more time real quick? Yeah, so the question is, do we look at soil solution chemistry? Besides just DOC, we did not do anything more of describing what that DOC was, but we do some EAMS work, that electron emission spectroscopy work. We did not do it on these ones, just also because, when we did this 2.5 years ago, that was something that we weren't doing. But I think DOC in this case, based on the microbial biomass that DOC supported, we can kind of assume that it was probably more phenolic, more recalcitrant DOC than normal. Okay, thanks.